Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 70th seminar, and I call it a University of Washington Day. There are many universities with Washington in their name, but University of Washington in Seattle is the best one among those, and the special one to me. By the way, I'm not affiliated with University of Washington, so I am not advertising it, or I have no conflict of interest. In fact, this SynBYSS seminar series started in 2021 thanks to University of Washington Chemical Engineering Department, which began an amazing seminar series called Distinguished Young Scholars Seminar in 2011 summer, 10 years before SynBYSS started. The department's pioneering idea of inviting young speakers to the chemical engineering department for a seminar was a new concept at that time. I was lucky enough to be invited and start the inaugural seminar series as the very first speaker, and that actually inspired me to this seminar series. I wanted to do the same thing for young generation. And in 2021, I started the SynBYSS seminar series. So I tremendously admire the vision of the department professors, including today's pioneer speaker. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor François Baneyx. He is, of course, a professor at University of Washington and the vice provost for innovation. He is one of the kindest professors whom I remember. When I was nervously giving my talk in 2011 in his department, I got a very difficult question and direct comment from a non-bio professor. Professor Baneyx helped me answer it and even defend for me. He may not remember it, but I learned from this experience that simple, kind word and caring heart could lead to big impacts on people. So thanks to him and my great experience during that visit, I made my mind and started this seminar series in order to do the same, but on the global scale to be novel or differentiate from their pioneering initiative. François, I really thank you for your time and for huge contribution to the scientific community and for my unforgettable memory. And the virtual podium is all yours. And thank you so much.
Thank you very much, Teosok, and thank you for the invitation. I did not remember that question, and I'm glad to hear that. And most of the professors at the University of Washington are actually very kind in their questions. So, I'm hoping that this was not a bad memory for you. I'm going to speak and share with you some insights and some mini epiphanies that I had through my career. It's not going to be a research talk. It's going to be very short, and I really want to have a good platform for Yana as she follows me. But let me go ahead and share my slide because I think this is something that matters. When I was first starting as a young assistant professor, I was still a postdoc at the time. David Goodsell published a really interesting papers that really changed my mind about how we're doing things and how we envision the cell. This paper was in Trends in Biochemical Sciences. This was in 1991. And what he did is that he basically said, well, what does the interior of a cell actually look like, right? We have all this information about dry cell weight, about proteins, about DNA, and so on and so forth. But what does it really look like when I'm trying to pack all these elements within a cell envelope? And this is one of the first picture that he produced, with ribosome proteins, mRNA, nucleic acid, and so on and so forth in this little 100 by 100 nanometer square of material. And here in this tiny little circle, you can see all these individual water molecules and small molecule there. And you can see the extraordinary packing and molecular crowding of the component. And David, being an artist, rendered all this stuff on watercolors on the paper. And I'm sure that many of you have seen his drawing of proteins in the protein data bank. And if you have not, please go there because they are fantastic. But this is giving you an idea of what is going on in the cell. This is not a sac that is full of fluid. This is full of material. There's an extraordinary amount of molecular crowding. And it is a miracle, actually, that life exists under these conditions when you think about it, that protein fold, that protein traffic, and that they go to different locations. And this is really the topic that I was interested in at the time, thinking about how molecular chaperones and foldaces are interacting with some of the nascent polypeptides that come out of the ribosome and help them either fold into the cytoplasm or maybe perhaps reach the inner membrane, go into the periplasmic space, fold there, or maybe, come out of the cell and be excreted for different purposes. And having this, in context was really extraordinary. And it was one of these mini epiphany. And I think that it has influenced a lot of the things that we are still doing, thinking about these type of issues. Cells are complex and life is very complex and it works very well and it operates within a couple of KTs. Now, a second epiphany that I had was when Helen Sable published this animated structure of the Groyel system on which we had been working on for a number of years. And this was built from cryo-EM data, basically reconstruction of the Groyel double stacked ring right here, and what it would look like when ADP or ATP binds to these different oligomers and induce these conformational changes and induce the allosteric switching that drives the mechanism of action of Groyel. And you can see even here, Groyel capping the protein and defining the cavity in which protein folding is going to take place. And that was extraordinary. Not only do you realize in a fail swoop way, what oligomerization can do for you, but what this marvelous molecular machine look like. And again, this was something that was a defining factor in how we approach things in the future, thinking about molecular machine functioning in this complex environment and thinking about assembly and self-assembly in particular, giving rise not only to complex protein with complex function, but other features such as the flagella that you can see represented right here. And now another thing that happened to me, which was, more in the late 1990s and perhaps early 2000, was the realization that inorganic marvelous structures can be formed biogenically. So you can see diatom skeletons here, going through one another, and you can see that these structures are absolutely fantastic. This was nothing new to a lot of people in the field. These structures had been observed by microscopy as early as the mid-1800s, but it was new to me, and just realizing that this could be achieved, by an organism and by protein, again, lend a new direction to our research and this interest in the bio-nano interface, in the interface between abiotic and biotic type of system was really important in driving our research. So what I wanted to showcase right here is that this type of epiphanies, this type of realization will happen to you over your career, and follow through and lean into them, because they will drive a lot of the things that you do. So keep an open mind and embrace this beautiful structure, this beautiful molecular machine, and the complexity of life. And I want to close by showing just one table of content figure of a paper that we published a couple of years ago. I'm not showing this for the science, I'm showing this for the fact that this stems from a very deep integration of skills and very deep collaborations. And this is what is going on in science today. We can still do things independently, but where we are going to make the most difference is by combining our respective skill sets and integrating them so that we can move forward. And what this shows you right here is that, for example, the progress that we have seen in molecular dynamics, where we used to be able to simulate things for a few nanoseconds, and now we can go for hundreds of nanoseconds, to maybe microseconds. And these are MD simulations that were conducted by Jim Fenner at the University of Washington, based on the starting structure that were obtained by Rosetta, created by David Baker at the University of Washington as well. And these two folks are part of the center that I direct right now. But we were looking at the interaction of these small peptides with inorganic surfaces. These happen to be silica surfaces. And this was only possible because of the progress that we have seen in computation over the past, let's say, 10 years. More importantly, this marvelous structure that I have seen of Gruyere that we obtained with great difficulty with cryo-electron microscopy and image averaging in the mid-90s have now been commoditized. You have the ability to do AFM and to actually identify single proteins here, self-associating or being independent right here, and connecting this to macromolecular outcomes. These images were taken by a colleague, Jim DeIorio, at PNNL, and these are liquid cell AFM, but they are pretty routine as well. So only by using the combination of these different expertise were we able to move from really molecular and atomic details all the way to a macromolecular behavior, in this case, a particular adhesion behavior. So I wanted to put this in front of you to get the ball rolling and people really thinking about what it means this day to conduct research and the power of working together towards a common goal. And with that, I am going to yield my time back to Tae-Suk so that Jan can go ahead and tell us her story. We look forward to it.
Fantastic. I absolutely agree with you about importance of collaboration. And, I actually always remember one African proverb that says, if you want to go faster, just go alone. If you want it to go far, go together. So that is absolutely, brilliant advice to young people. Thank you so much. That's brilliant. And by the way, the question I got was not, nasty one. That's a very helpful one, because that is from chemical engineering professor perspective. I thinking about things bioengineers perspective. So that opened my eyes, because that helped me later on for the interview process. Because if I didn't think that one, I have bombarded with some question I never expected, right? So that was the very good experience. I still remember because of that.
Fantastic. I'm glad to hear.
Yeah. Okay. So now the main speaker of today with a longer introduction. Dr. Yan Zhang is a presidential postdoctoral fellow at the California Institute of Technology, Caltech, in Professor Richard Murray's group. Her research interest is in constructing synthetic cells as the next generation chassis to implement engineered biological functions. Yan recently completed her PhD in chemical engineering at Georgia Tech, where she explored various interfaces to advance point-of-care biosense diagnostics. She earned her bachelor degree in chemical engineering from Cornell University, where she was first introduced to synthetic biology in Julius Lux's lab that focused on developing RNA-based genetic regulators. Outside of research, she is actively involved in leading science outreach programs, encouraging more participation in synthetic biology at the International Genetically Engineered Machine, IGEM, and Engineering Biology Research Consortium, EBRC, my favorite nonprofit organization, where I am also a council member. She is a true rising star, and if you are a professor who is planning to hire a future leader, I highly recommend you should act in advance before everyone sees her potential and capability, and she joins somewhere before you wanted to take her. Yan, thanks so much for your time today. Please take it away. It's all yours now. Thank you.
Well, thank you, Tae-sok, for that wonderful introduction. And also just like providing this platform where young speakers and early career scientists can share their work. And let me begin sharing my slides. So is it visible?
Yes, yes. Go ahead.
Oh, okay. Awesome. All right. So as Tae-sok introduced me, I'm currently a postdoctoral fellow at Caltech, but the research being shared today is actually my PhD thesis work that I have done at Georgia Tech with my thesis advisor, Mark Stasinski, where we developed protocell arrays for simultaneous detection of diverse classes of analytes. So why was I really interested in detecting diverse classes of analyte? I'm going to use the recent COVID-19 pandemic as an example. And I think one of the lessons we've all learned during COVID-19 pandemic is that our demand for affordable and accessible diagnostics far exceeds the current supply of these diagnostic devices. And to meet the demand for these diagnostics during COVID, we have ramped up university facilities to provide large-scale surveillance testing and symptomatic testing for students, faculties, and staffs. And even at the community level, we have these neighborhood testing pop-up sites. So people in the community can just walk in, drop off their samples, and get their results in a day or two. And even for people in communities that don't have access to university labs or neighborhood pop-up sites, at-home diagnostics are being developed so they can be ordered from supermarkets or online for these diagnostics to happen at the comfort of people's homes. And if we can make this happen for COVID, can we make this happen for other relevant diseases like micronutrient deficiency, heavy metal poisoning, and also pathogenic infections? A lot of these tests are still being done in university hospitals or centralized laboratories where access to these kind of testing is limited and also cost prohibitive to communities that are most vulnerable for these diseases. So how can we advance point of care diagnostics toward all analytes to detect all diseases? This question has been the biggest motivating factor of my doctoral research at Georgia Tech, where I try to develop different point of care diagnostic devices using cell-free biosensors. So why use cell-free biosensors to develop these low-cost field-deployable diagnostics? Living cells can already be seen as experts at carrying out different sense and response functions. They're almost like these little intricate molecular laboratories, constantly sensing and regulating analytes across different molecular classes, from ions to small molecules to nucleic acids to proteins, all of the time. And cell-free system harness these capabilities of living cells at carrying out genetic programs and sensing different analytes without having to keep these cells alive. And this really improves the portability and applicability of these biosensors. And all we need to do is just grow up a bunch of cells, lyse them, and harvest their core protein machinery to carrying out these sensing functions into a tube of extract. And into this tube of extract, we can add customized genetic circuits or biosensing programs to allow sensing occur toward different analytes to detect different diseases. And our lab at Georgia Tech, the Szynski lab, took advantage of cell-free biosensors to develop a colorimetric micronutrient biosensor. So I was very lucky to work with a senior graduate student at the time, Monica, and also an undergraduate student, Paige. And together we load a sensing program that detects the amount of serum zinc available, and the zinc available regulates how much LAC-Z enzyme is being produced, which then regulates how much colorimetric output the sensor outputs, and we're able to infer the zinc concentration in human serum by reference it with a established set of standards. So by matching the color of different cell-free sensing reactions to the standard reference reaction, we're able to identify whether the patient is zinc deficient or not. And we show that this approach is also applicable to quantify RNA concentrations in the field, with emerging applications to identify and quantify viral load concentrations and patients' response to antiviral treatments. So being really inspired by this equipment-free point-of-care diagnostic work, I was able to continue to work with Paige and also an additional undergraduate student, Max, where we switched up the colorimetric output our sensor was generating with a sugar molecule. So LAC-Z, instead of generating colorimetric outputs, can also cleave sugar molecule lactose to produce glucose. And the glucose being produced can directly be interfaced with a personal glucose monitor. So we're able to achieve digital quantification of human zinc concentration. And we no longer have to look at the color and try to determine whether this looks more orange than it is red for inferring zinc deficiency. And Max also showed that in addition to detecting micronutrient, we can also detect RNA samples to identify pathogenic infection. And that was really exciting. But what we noticed when we're developing these point-of-care biosensors and interfaces to implement them at point-of-care settings is that a lot of the biological samples that we use to detect different diseases have multiple relevant analytes for detection. But each of the biosensing tests we developed can only detect one biomarker at a time. Can we flip this around so we simultaneously detect multiple relevant analytes from the biological sample using just one sample? And with our collaborators at Georgia Tech from the Takayama lab and also the postdoctoral fellow there at Tai, we show that this is possible using aqueous two-phase systems. Aqueous two-phase systems, the most prominent example of such systems would be the oil and water phase separation, based on hydrophobic and hydrophilic interactions. And the same type of phase separation also occur in the polymer world. So when we mix different weight ratios of polyethylene glycol, PEG, with phycol or dextran, these polymers will spontaneously phase separate because of hydrophobic and hydrophilic behaviors. And at the interface of these polymer phase separation, we see really interesting biomolecular partitioning properties going on, where more hydrophilic molecules like nucleic acid or protein would selectively partition into and stay localized in a relatively more hydrophilic phycol or dextran phase. And we thought this could be a really cool strategy to create phase-separated biosensors that can coexist but remain spatially separated in the same PEG environment. So individual separated phycol or dextran phase would contain the cell-free biosensors, almost forming a protocell. And when the entire PEG environment contains the analyte, a mixture of analytes that we want to detect, these analytes can then all enter individually phase-separated protocells to activate their respective sense and response program. And by tracking which location of these protocells lights up in a multiplexing well, we're able to identify which analyte is present and which analyte is absent. And to test this idea, we first need to characterize which two-phase systems can be interfaced well with cell-free biosensors. So we chose the two candidates of polymers, phycol and dextran, and then to characterize their ability to work with cell-free biosensors, we simply vortexed our cell-free protein expression mixture into each polymer solution. And then to create these two-phase separations, we first load the bulk phase of the well with polyethylene glycol and also the energy buffer for cell-free reactions. And then to the loaded PEG environment, we add individually prepared cell-free protocell mixture, and we incubate them for a period of time before we image them for fluorescent results. So here is a representative plate map for a 4-plex protocell system, where at this diagonal, we have empty cell-free reactions encapsulated in different polymers. And at this diagonal, we have cell-free reactions expressing a green fluorescent protein. So we're able to track whether the protein is being produced and localized in the protocell compartment that they're in. And using a customized 96-well plate, we have 4 imprinted microbasins that house these phase-separated protocells. And we show that for phycol-based cell-free reaction protocells, it actually produces the highest green fluorescent protein signal and the lowest leak to surrounding protocell environment. And that was the system that we chose going forward. And having a protocell array environment set up, we thought it would be very exciting for us to first show that we can detect multiple model small molecules. So in the same 4-plex sensors, the 4-plex well environment, we embedded 2 different model small molecule biosensors. One is the IPTG sensor in this purple well, where the addition of IPTG de-represses the LACI promoter and produces GFP output. And in the yellow well, the addition of arabinose would activate GFP expression from its respective promoter. And we show that when none of these small molecule inducers were added to the surrounding bulk PEG environment, there is no activation of these sensors. And when arabinose is added to the PEG phase, we see strong and distinct GFP production coming from the arabinose sensor only. And similarly for IPTG, only the cell-free protocell that carries the IPTG sensor produces GFP. And when both small molecules are added to the environment, both cell-free biosensors produced signals responding to the small molecules added in the bulk phase. And that was really exciting. And we thought, in addition to small molecules, can we detect something different like nucleic acids? And using Toho switches that were developed by the Collins lab at MIT, we showed that, so for Toho switches, it detects RNA sequences, and without an RNA trigger, the mRNA would fold into an inhibitory structure that represses downstream gene expression. And in the presence of an RNA trigger, it will unwind this inhibitory hairpin and produce GFP as our signal. And we implemented these Toho switches in our 4-plex system and embedded already characterized RNA triggers B and RNA triggers H as our inducer to activate these Toho switch circuits. So when RNA triggers are being added to the well, we see differential activation of their respective sensors. And we show that when RNAH is being added, similarly, we see dose-dependent activation of these GFP signals. And when RNA B and H are all added to the environment, we see strong and distinct activation of both Toho switch sensors. And that was very exciting for us. But we also see that when both RNA triggers are added, there is actually reduction of signals in this co-expression case compared to single expression case. So when we try to characterize this, we saw that it was just a very sequence-specific interaction beyond between these two triggers. It was just tough luck that we picked these two triggers for multiplexing, but these interactions don't happen with other random trigger sequences. So it wasn't something that was wrong with this multiplexing platform. It was just sequence interactions in between these switches. So that was really cool. And we thought it would be pretty unrealistic for us to try to detect micromolar concentrations of RNA triggers in clinical settings. So we thought, is there a way for us to improve the sensitivity so we can detect these nucleic acid signals at higher sensitivity than micromolar? Can we push it into nanomolar ranges? And we showed that this is possible if we add linear DNA coding for these Toho switch trigger expression. So in the bulk phase, the PEG phase, instead of adding RNA triggers, we can just add DNA coding for these RNA trigger expression. And we show that when we add DNAs into these PEG environment, we can now start seeing these strong sensor response at the nanomolar range. And they're also specific to the signals that's being added. And at this point, we're like really excited about how our platform can detect both small molecules and nucleic acids. But they're all very well-established model systems. Can we detect something that is clinically relevant and also coming from diverse molecular classes? So we created this contrived diagnostic panel consisting of a zinc sensor, a vitamin B12 sensor. There are 2 relevant micronutrients. And we've also developed Toho switch sensors that can detect pathogenic E. coli by targeting the Shiga toxin sequences that are present in pathogenic E. coli. And to further mix things up a little, our entire panel consists of an ion that is zinc, a small molecule that is vitamin B12, and RNA triggers coding for Shiga toxin 1 expression, and also DNA coding for Shiga toxin 2 trigger expression. And we show that when none of these inducers are being added to our multiplexing environment, we don't see activation of our sensor. But then when zinc and DNA is being added, our sensor array is able to pick up these ion and DNA signals and produce distinct output related to these signals. Similarly for vitamin B12 and also RNA trigger. And then when all of these inducers are being added, all these sensor lights up, indicating detection of molecules across from ion to small molecule to nucleic acids. So we've demonstrated here that we can multiplex analytes across diverse classes of molecules, but can our platform actually be interfaced with samples that are clinically relevant? And one sample we think of that is being clinically relevant would be human serum that is being used in many diagnostics. And we show that our sensor array can also take 20% of human serum and still produce these distinct sensor outputs using fluorescence response. And we try to use higher concentrations of human serum, and that was not possible because human serum would generate these protein precipitate in the PEG environment. So 20% was the highest concentration we can use in the testing setting. At this point, we have shown that our multiplexing platform can detect diverse classes of analyte and also be interfaced with a clinically relevant human serum sample. But can we actually make this sensor array accessible for point of care deployment? And these platforms can be shipped to and used in environment that need them to function. And when we think of point of care deployment, it would be very unrealistic to think like mobile clinics will have fluorescence imagers to detect GFP. So the first thing we did, moving toward point of care deployment, is to actually replace these fluorescent reporter with a pigment converting enzyme that can produce visible results. So we switched GFP with enzyme LAC-Z, which we have shown in previous works can cleave a colorimetric substrate to produce a visible color output. And although this LAC-Z cleaving CPRG to produce visualizable CPR purple color works, CPR is a small molecule and it would diffuse out of these aqueous two-phase systems. And even though we can see distinct color separations in 30 minutes, if we have sensor responding at later time points, these small molecule colorimetric signals will start to diffuse out. So if we have sensors responding at different time points, we may lose these resolutions of which sensor got activated and which sensor did not. So we switched from CPRG to an older colorimetric system LAC-Z was used for, the X-scale to a blue precipitate system. And it was really cool because these blue precipitate actually stays localized in these phase-separated protocells. So even when using X-scale as a colorimetric output is 30 minutes slower, we're able to see more localized pigment generation, which can be used for result identification at later time points. And after establishing this colorimetric output, we thought it would be really cool to establish some kind of testing panel to have visually detectable multiplexed analytes occur in the same testing chip. And to do this, we created a panel of different microbiome targets. So using the pathogenic E. coli Toho switches that we developed before that identified Shiga toxin 1 and Shiga toxin 2 sensors. And we've also thrown in another opportunistic pathogen in the gut, B theta iota micron, which would turn into a pathogenic species when during the event of pathogenic E. coli infection in the gut. And we show that for this mini testing panel, when no DNA triggers coding for RNA trigger expression are present, none of our sensors produce any visible color output. But when B theta triggers are being added to the environment, only B theta sensor creates a colorimetric output that's visible in just 1 hour. And the signal is more pronounced in 2 hours. And for when we add Shiga toxin 1 sensor, only Shiga toxin 1 sensor will produce a colorimetric output. And when Shiga toxin 2 is added, only Shiga toxin 2 sensor produce colorimetric output. And when all of these DNA triggers are being added, all of these sensors produce visible colorimetric output in 2 hours of assay time. So this shows that we can use these protocell arrays to identify a panel of pathogenic infections without any aid of instruments for visualization. These can be interpreted using our eyes. And after establishing this platform for colorimetric output, we thought, can this platform actually get to the field? A huge thing for a lot of diagnostics and even therapeutics is that they require cold chain shipment. And that logistic involved in shipping these diagnostic or therapeutics out using cold chain is actually what is preventing point of care diagnostics being point of care and accessible. So we showed that we can freeze dry the components of our multiplexing platform, the PEG bulk phase, and also our Protocell array system into a dehydrated pellet form. And these pellet forms would be shelf stable at room temperature. And when it's time to carry out these diagnostic reactions, we simply can just add water to rehydrate the PEG bulk phase and use the rehydrated PEG bulk phase to hydrate our Protocell array system. And all we need is to incubate the rehydrated reaction for 3 hours at 37 degrees, and we see restored Protocell array formation and also sensor output without losing sensitivity to the signals. So to recap my PhD work, I have introduced our Protocell array system where we embed different cell-free biosensors carrying customized, unique biosensing program into phase-separated droplets formed by polymer aqueous two-phase system. And then using these phase-separated protocell biosensors, we show that these protocell biosensors can produce analyte-specific activation and reporter production to signals added to the external PEG environment. And for these protocell array sensors, we can simultaneously detect diverse classes of biomarker molecules in a clinically relevant context using human serum. And lastly, we have demonstrated a path toward a field-friendly diagnostic application of our protocell array at becoming a diagnostic panel for screening different pathogenic infections and potentially more. So none of these would be possible without my thesis advisor and also our wonderful collaborator. And just going with the theme of like science is not possible without collaborations, right? This is exactly that, right? Because how we came up with this project idea actually came out of a seminar talk that Shu Takayama was giving at Georgia Tech. And he was giving a talk on antibody-based multiplexing. And we thought, wait, antibodies or proteins, they can be embedded in these polymers. Cell-free biosensors are protein too. Can we also multiplex cell-free biosensors using that? And Mark, just being an incredible cheerleader of my PhD work, told me that like I should talk to Shu and get this project started. And here we are having this like really amazing work output just coming out of a seminar and a very like cheerleading type of advisor. So I couldn't thank them enough for giving me the creative freedom in designing this project. And also my collaborators, the postdoc at the Takayama lab, Tai, and also the senior graduate student in the Szynski lab at the time, Monica, and my wonderful, wonderful undergraduate mentees. And I would also like to thank my funding sources and also labs at Georgia Tech and also Northwestern to providing the facilities, equipment, and also the cell strings to do a lot of these cell-free biosensing work. So I guess at this point, thank you for listening to my presentation, and I'll be happy to take any questions.
Fantastic, fantastic. I love it, love it. So, Francois, do you have any question, comment?
I do. First, it's fantastic, great presentation. Yeah, I really appreciate it, and it's beautifully done. I had, I assume that you're using cell lysate that you put into the cycle in addition to your sensing elements. So, my question was related to the diffusion of small molecule into the PEG phase, and how this is affecting things, in particular your efficiency of translation, and how your signal is going to be maintained for a long period of time. And conversely, whether or not you have experienced different diffusional limitation of different, analytes in the PEG phase, and that could affect, the time to response that you see.
Yes, so I think the first question is the diffusion of small molecules. I think let me go to the relevant slide. So in characterizing a lot of the small molecule biosensors, we actually put a set amount of time on the reaction incubation. So a lot of these protocell array results are imaged at the 3rd hour. And at the 3rd hour, there is enough equilibration going on. So we assume all the small molecules have went in to these phase-separated protocells. And it's because of the specific time that we are looking at, we're not able to see if a specific analyte, like arabinose or IPTG, are going into these polymers faster than the other. And that is just for small molecules. But for nucleic acids, we see the structure of nucleic acids actually strongly influence their partitioning and concentration effects in these protocell arrays. So what's really cool is we see that for RNA triggers that have very defined structures, like if they fold into a hairpin, they would partition into these polymer environment way faster than RNA that don't have defined structure. So you would imagine like a very flexible RNA molecule where it's just ACUG on repeats. So it could be a really interesting area to look into if like specific structures being added to RNA can allow these RNAs to go into these sensors faster and becomes more concentrated. But so far for small molecules, we don't really see a diffusion impacting their entrance into these sensor, just because we're looking at these response in 3 hours. But if we were to look at these time slices at 30 minutes, 1 hour, maybe we'll have better resolution at their diffusion properties.
Yeah, and the point that you make is a really important one. I think that because you have flexibility in printing, these protocells, you could actually exploit this diffusional advantage by actually having protocells of different shape that will affect how the various materials are being sensed and when they're being sensed.
Mm-hmm.
Great work, thank you.
Do you have any other question?
Not right now, thank you. I really appreciated the talk. It's great, very exciting. Going to relevant concentration of analyte in clinically relevant sample is of course a challenge, but you're on your way there, so it's really good.
So yeah, Jan, I have multiple questions, but I ask some simple question first, and then we discuss later. So This is fantastic. You tested multiple different things, and then the real application relevant condition. That's fantastic. And I believe your lab probably already done before, but could you kind of comment on, selectivity or specificity of, for example, I don't worry about Tor switch in terms of specificity, but how about specificity for, zinc sensor and some other sensor? I may miss that part, but I want to ask that question.
Yes, so that's a great question. So as we were developing zinc sensors, in the previous work from Monica, it was actually a reviewer's response, like, hey, you're developing these zinc sensors to detect clinically relevant micronutrients, but a lot of these metal sensors have promiscuity responding to metals like copper and other heavy metal analytes. And we see that for this particular transcription factor that we're using, ZNTR, this specific one that we use is quite selective for zinc. And also in human serum that we're detecting, zinc concentrations are in the range of micromolars. So I think also like an argument for selectivity is like, if there are heavy metals like copper that are present in human serum at micromolar levels, like the person would probably already die of heavy metal poisoning. So we show that like ZNTR is actually quite selective for zinc in this specific protein that we chose, and also the concentration that this transcription factor is responding to, we shouldn't expect other heavy metals to contaminate and convolute the results.
I see, thank you. I guess, we only have 1 more minute, so I will close and then we'll discuss more. I have a couple more questions. All right. So thank all for joining and staying today. We'll meet again on March 9, Thursday, the same time, the same Zoom link. We'll have Professor Chang Lu at UC Irvine and Professor Anam Glasgow at Columbia University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face if you wish. And thanks. I will stop recording. Thank you. All right.